Okay, so we are going to go ahead and get started. Hi, everybody. Good afternoon. My name is Anjali Gujral, and I'm an assistant professor at the Emory University Global Diabetes Research Center. And I am delighted to introduce our speaker today, Dr. Lisa Staimez. Dr. Staimez is an associate professor at Emory University at the Global Diabetes Research Center. And Dr. Staimez received a bachelor's degree in molecular and cellular biology and her master's in public health from Yale University, studying chronic disease epidemiology. And Dr. Staimez also spent some time working at the Center at the CDC, Center for Disease Control and Prevention. And since she has been at Emory, Dr. Steinmetz has been really spending a lot of time looking at type 2 diabetes pathophysiology and really working to identify markers of early stages in pancreatic islet cell dysfunction. And Dr. Steinmetz really works at the intersection of epidemiology, biology, clinical science, multiomics, population health, nutrition. So she works to combine all of her background and all of these really interesting interests into generating new evidence for mechanisms of identifying diabetes in the hopes of informing and developing innovative pathways for diabetes prevention and control. So with that, again, it is my pleasure and honor to introduce Dr. Lisa Staimez.
Thank you, Dr. Gujral. I appreciate that kind introduction. And it is great to be here with you all to talk about what many of us find important, which is trying to solve the challenges that are brought forth with the growth of type 2 diabetes. So I will advance my slides here. Okay, so the objectives for me and for you for this talk will be to review a little bit of the premise behind the work that I'll be talking about in this presentation on some background related to type 2 diabetes. My focus will be on youth, but much of what I will be saying also applies to adult populations. And then I will kind of dig a little bit into what methods we have available for population screening and how we might challenge those methods. And then lead into what emerging research is at play right now for screening and also for identifying mechanisms of type 2 diabetes that will make a difference in outcomes and changing the natural history for diabetes in youth and adults. So just a little bit about me. And Dr. Gujral did touch on this a little bit, but I come from a foundation of applied practice of global public health work, and that is based in formative work for me in my identity as a professional at INCAP, which is based out of the PAHO, part of the World Health Organization, and also the CDC. And those were places where I got to see how public health and programs and governance works. My training is in epidemiology and nutrition, and I currently work in the Emory Department of Global Health, and I'm a faculty also in the Nutrition and Health Sciences program. And my research focus is in diabetes epidemiology and in pathophysiology, and I have the pleasure of doing this work at the Emory Global Diabetes Research Center. And so some of my work is focused on diabetes in youth. I first became interested in doing this kind of work because of the kind of nutrition interventions that I was working on in Central America from many other years ago, where I got to see the nutrition transition at play with both underweight and overweight starting to impact low and middle income countries. And so the projections for type 2, for diabetes in general, for youth in the United States is not so promising because we're still going to be seeing an increase up from latest numbers that were captured in 2017. So this is all from the Search Diabetes in Youth study. And what we can see here is that there will be a growth. The blue bars represent type 1 diabetes, and the orange bars represent type 2 diabetes. And these are cases per 1,000. You can see that type 1 diabetes is continuing to increase, and type 2 diabetes is actually increasing all the more. Actually, that increase is 700%. That's the projection by 2060. And now what we know from more recent trials that have included both adults and youth, we understand that youth actually progress through the natural history of type 2 diabetes more rapidly than adults. In the absence of pancreatic autoantibodies, there is this silent deterioration of pancreatic beta cells. This happens sometimes without any symptoms, and the pancreatic beta cells are trying to compensate for stress that they're experiencing, and then eventually they fail, and that's what leads to overt hyperglycemia, diabetes. So for youth, the decline in beta cell function is 20 to 35% per year, whereas in adults, the decline is 7 to 11% per year. And also 52% of youth are not responding to metformin therapy compared to 12% of adults. And so we are trying to understand, why is it that there's this kind of accelerated time course and experience through the natural history for youth. And so far, we don't have a lot of answers, but, there's been a lot of signal related to adiposity and ensuing insulin resistance. This picture becomes a little more grim when you consider type 2 diabetes complications. And so what we see here is some data from the Today study, which originally enrolled 10 to 17-year-olds with diabetes, like new diabetes within the last 2 years of diagnosis, and who were overweight. And there was a treatment trial phase between 2004 and 2011. And then there was an observational follow-up phase from 2011 to 2020. And these are all kids negative for antibodies. So they don't have, it's presumed that they don't have type 1 diabetes. And after about 10 years, just over 10 years, the Today study was able to show what the incidence of complications are. You can see cumulative incidence here for hypertension, kidney disease, 50%, 55%, and cumulative incidence of dyslipidemia, 52%, nerve disease, 32%. So this is not a positive picture, especially for people who are in their most productive stages of the life course. And so coupled with data from the Search study, which also had an 8-year they also published data from an 8-year follow-up period. 70% of youth with diabetes, with type 2 diabetes, developed a complication by the age of 21. And they recorded this related to diabetic kidney disease, retinopathy, peripheral neuropathy. Even in this study here, if you look at any one complication, if what is the cumulative incidence of any microvascular complication, it was 80% during this follow-up time. So the risk factors for developing these complications include being a minority race or ethnic group, having a high glycated hemoglobin level, having low insulin sensitivity, having hypertension, and having dyslipidemia. That those are the things that were related with higher incidence, or with risk. So what I think is particularly both or I think there's an opportunity is where we consider the opportunity for in this dire situation is where we consider age of diagnosis. And this is a study in adults, but I think we can apply what we learn and extend this to a little bit earlier in the life course. So this is an analysis of adults who this was a study that integrated data from 26 other studies into a meta-analysis and covered 30 countries and over 1.3 million people. And they did this random effects meta-analysis and pooled odds ratios. And let me move this bar. Okay, sorry about that. And so they wanted to see what is the association of age at diabetes diagnosis. What is the association of that with all-cause mortality, macrovascular disease, and microvascular disease? And what you can see is that the age at diabetes diagnosis was inversely associated with each of these factors, and that each 1 year increase in age at diagnosis actually was associated with a 4% decrease in all-cause mortality, 3% decrease risk of macrovascular disease, and a 5% decreased risk in microvascular disease. So this is really promising and also shows some urgency that we need to address this, especially for the younger folks that might be younger adults, might be youth, adolescents. If you're in the younger part of your life course and and there's some signs that things could be going in the wrong direction related to development of type 2 diabetes, that it's very important to intervene because the intervention will have a positive effect in diminishing these kinds of long-term outcomes. So in the Today trial, which was a trial of youth, those who succeeded in the trial had better beta cell function, better glycemic control at baseline. So I think the idea is that where we can intervene and improve beta cell function right off the bat, that's where you have better long-term outcomes. And so our opportunity is to preserve beta cells during early stages of decline prior to the loss of glycemic control. So what can we do to prevent beta cell decline? Broadly, I think of this in two categories. One is screening, where you take measures, whether they're associated with the disease course, whether they're actually involved, whether those measures are involved or not involved, taking measures that have high sensitivity and specificity to predict, and using those as screening tools. Then there is the development of, and we have existing screening tools, and I'll talk about those briefly, but then there's a development of innovative screening tools that we could look towards. And then secondly, what we can do to prevent beta cell decline would be to identify new mechanistic targets and develop interventions around them. So I'll start with the focus on screening for now. So beta cell function specifically is not routinely measured, but it is the most intimately related to onset of type 2 diabetes. Common clinical measures are used as surrogates, and many of these are downstream metabolic features, and those include glucose, those include HbA1c. They can also include insulin, although less frequently measured. And there are some caveats to insulin in that there are competing metabolic processes that may impact how insulin is measured, which could include, for example, insulin clearance. The most common screening measures were developed with adults in mind and may not be specific enough to the unique pathologies we see in youth, such as rapid beta cell decline. And here's an example of the tools that we use today. So we have the fasting plasma glucose, 2-hour plasma glucose, HbA1c, random glucose. And you should note that not all of these tests necessarily detect diabetes in the same individual. So some individuals might have diabetes by this measure, but not these, as an example. Or some might have diabetes based on HbA1c, but not glucose levels. Also, these tests are applied equally, or meaning the cut points are the same, regardless of whether someone is low risk or high risk. So there are good reasons for using these variables because they are easy to scale up to the population level. And here's an example of a screening plan developed for the integration with a microsimulation model that we were working on here at Emory with CDC and our partners RTI for some modeling long-term outcomes in people with type 2 diabetes. So ultimately the screening variables may not be enough to reflect something as specific as pancreatic beta cell function, which is the root problem of type 2 diabetes. And so a question that I ask is to what extent we're classifying individuals who have high risk for beta cell decline on pathways that don't lead to treatment. For example, you may have individuals who are false negatives for diabetes, and is there a way for us to, and then they may end up without having any intervention. So what do we do about those individuals? Can we refine this further? So HbA1c is another measure that we use. It's the cut points are uniform, for the population, but HbA1c does not uniformly reflect blood glucose levels across subgroups. And we would expect, based on glucose levels, that predicted A1C would be different, actually. This happens, this kind of mismatch happens in certain segments of the population, and I'll give you an example of that. We did a study of NHANES between 2005 and 2014 data of which is representative of the United States adults, and we found that the optimal thresholds for HbA1c was higher in Black participants, which is this row, versus white participants, which is this row. So here you can see the little red star is where that denotes Youden's index, which is a measure of where specificity and sensitivity are maximized in the classification of so in this case, we're classifying diabetes and we're comparing the classification. We're including everyone in this, everyone else in the population. So Youden's index was greatest in Blacks at HbA1c of 6%. But in whites, Youden's index was greatest at 5.7% in whites. And then when you exclude people who have normal glycemia and you do the analysis again, this is, you see the Youden's index for Blacks is at 6.2% A1C, whereas in whites, it's 5.7%. So the difference between these two is about 0.5% A1C. And what we know from other studies is that is enough, that difference, 0.5% in A1C, is a clinically significant difference that could lead to the misdiagnosis or inappropriate management of, and actually what we did in this analysis, we quantified that and we showed that 30 million Americans may be misdiagnosed or given it different management than what they would possibly need. Here is a different analysis where we looked by age groups. We wanted to see, does this phenomenon can we see this in young people as well as older people? And so here is, this is an analysis of an index called the hemoglobin glycation index, which is a very simple observed A1C minus predicted A1C. And the predicted A1C comes from, we calculated that based on glucose levels from an OGTT. So the vertical line here represents zero deviation from observed and expected A1C. And then bars to the left of the zero line suggest that A1C was less than predicted. The observed A1C was less than predicted. And then these bars here on the right show that they represent the observed A1C that was greater than predicted. And these numbers here above the bars is the percent of the US population. So the highest deviation are in this bright blue color. And so we show that 4% of the US population, among 12 to 17-year-olds, 4% of them fall into this really high deviation category, and as well as this 5% on the lower end. And then what we did was we further stratified by race. And this is where, it can be pretty striking. So you can see here, it's the 12 to 17-year-olds that we looked at here. We're only, this graph is only looking at these extreme ends. So the orange bars here, for example, these are the ones that had a mismatch of observed minus predicted of 0.5 or higher. And what you'll see here is that the prevalence is much higher in non-Hispanic Blacks compared to non-Hispanic whites. So HbA1c needs a little work. And so we wanted to know, okay, what else can we be using? And so our objective is to look at oral glucose tolerance. One of our objectives is to look at oral glucose tolerance tests and see how we can better adapt those to assess beta cell function, especially in young people. And there are these other indices like HOMA-B, which uses only fasting measures, the oral disposition index, which uses the challenge of the oral glucose tolerance test to create an index. And there are several others like this. And we were able to try to adapt the method for infants in a study called the ABC study, the Asian Indian beta cells in infant study, where our objective was not only to apply an oral glucose tolerance test for beta cell function in infants, but also to assess early life nutritional risk factors, specifically maternal nutritional status, measured as maternal BMI, and how that might be associated with insulin secretion in their infants. And we did, in the third trimester, recruit women who were pregnant, went back retrospectively to get their BMIs, and then got glucose and insulin levels from cord blood and dynamic insulin and glucose levels through an oral glucose tolerance test which was adapted just for the first 30 minutes of the test in their infants. And we did find that there were differences, by the way, with the maternal nutritional status, like what was the mother's BMI in her first trimester, and how was that associated with the infant's beta cell function. We found this U-shaped, using static measures of beta cell function, we did find a U-shaped association where beta cell function was higher in the underweight, the infants from underweight mothers and the infants from the overweight mothers. And these blue bars here were the same across all groups. This is the insulin resistance levels. So while insulin resistance was the same across these groups, the beta cell function by HOMA-B was varied. And then 7 months later, when we looked at the infants again, the levels of insulin secretion were sustained in the babies from the overweight mothers, whereas those from the underweight mothers, there was a decline in beta cell function. So the point really for me to share this with you in this talk is really more about how appropriate might this be for population scaling and what else can we do There's something useful that we've learned in this study, that there is in utero, potential in utero effect of nutritional status on the next generation's metabolic factors. But practical question is also, is this, how appropriate would this be for scaling up and using more widespread? And so that leads me to this next part of the talk, which is thinking about screening innovations. Those are presently being informed by global population studies, where new information about disease development are being identified to really identify that there's heterogeneity in what dominant pathways they are driving type 2 diabetes. So in our study of Asian Indian youth, for example, we identified early impairments of insulin secretion that were identified earlier in the natural history of disease than expected, and so suggesting that maybe insulin secretion is like very dominant pathway. And this is counter to older traditional paradigms of diabetes development. And then when we look, and this is very much what we see in our adult studies, like, for example, this study where we compared two high-risk populations of adults, Asian Indians and Pima Indians, who have high prevalence of type 2 diabetes in adulthood, but who that are very different in other ways. For example, anthropometric features, amount of BMI, et cetera. What we see here is really some striking differences. So you can see the solid lines are showing across the natural history of type 2 diabetes. They're showing the insulin levels at fasting for Asian Indians in the light green and Pima Indians in the dark green. And when you look at response to a stimulus like a glucose drink, you see that the response in the Pima Indians in this dark dotted line is much higher. The insulin levels that were produced were much higher in the Pima Indians compared to the Asian Indians. And so this has all been part of a foundation of work that's led to these hypotheses that maybe there are these subtypes of leading to type 2 diabetes, where one phenotype might be related to wide beta cell capacity and the ability to secrete insulin in a compensatory kind of way, and to be able to meet the metabolic demands of the body. Where a dominant form of prediabetes would be impaired glucose tolerance, and that obesity is a very central part of this paradigm. Whereas this phenotype, the second phenotype, would be based on this narrow beta cell capacity, where even slight requirements for insulin would cause strain to the beta cells, and driving normal glucose tolerance individuals into impaired fasting glucose, and then eventually converting to diabetes, even if insulin resistance is not that high. And so this is leading to many really interesting innovations in classification right now. This is a paper by Alkvist and colleagues. The authors were motivated by the observed heterogeneity of type 2 diabetes in classification. And so they did this data-driven cluster analysis in nearly 9,000 newly diagnosed diabetes cases from a Swedish cohort. And the clusters were based on 6 variables, based on antibodies, based on age at diagnosis, based on BMI, HbA1c, HOMA2b, and HOMA2IR. And using those data from those variables, they identified 5 clusters that they could replicate with differing disease progression characteristics and different characteristics related to diabetes complication, risk for diabetes complications. And I'll just bring attention to like one, which was an insulin resistant cluster. This is cluster 3. And this other cluster 2, which is an insulin deficient cluster. And so This has then been examined further. There's been many other clustering analyses that have been done, but in slightly different ways. This here is a slide from a systematic review where authors wanted to see whether the strategies that have been used so far to subclassify type 2 diabetes have been associated with high quality evidence, whether the results are reproducible, and which have been, linked to improved outcomes in patients if they're in longitudinal data. And did find 62 studies with complex stratification methods and reproducible subtypes for type 2 diabetes. So here's the ALKVIS clusters and the 22 publications that have used those. And it does need to be some work to look at reproducibility in a little bit more of a varied set of populations and different ethnicities, et cetera. But this is very promising for where classification of type 2 diabetes could go. Now, while in thinking about innovations to screening, epigenetics is one area of my focus. And that's because while genetic differences and genetic differences have been explored, but they are somewhat limited in fully explaining differences in type 2 diabetes risks. And so recognizing that perhaps epigenetics and the expression, the control of gene expression may be a really good place to look. Epigenetics is how genes are expressed or how they're turned on and off. It's kind of a element of regulation. And epigenetic markers can be stable through cell division. They can be stable through transgenerational inheritance, and they can persist even in the absence of conditions that established them. So there's some biological memory that can occur around them. And so because modifications to the epigenome are dynamic, and they're responsive to environmental exposures, and they're considered stable adequately for inheritance, the epigenome is a highly plausible source of glycemic variability across populations that would be worth exploring for screening and certainly for mechanistic understanding. And so one example of epigenetics is DNA methylation. And there are examples, or there's already a literature on how DNA methylation is associated with glucose homeostasis. For example, CpG methylation is associated with fasting insulin and with fasting glucose. The role of differential methylation in the crosstalk between adaptive immune system and glucose homeostasis has been documented in publications. And differential methylation can explain at least around 17% of the association between obesity and insulin, which is another important part of the picture. And methylation is predictive of increased risk of type 2 diabetes among some ethnicities versus others. So if there's a differential in risk between certain ethnicities, DNA methylation has been able to predict that difference in risk. The limitations, though, for DNA methylation are around the general bulk approaches of these kind of analyses, and which characterize average epigenomic features across multiple cell types that have vastly different function. Now, for the purpose of screening, that may be okay, although we haven't yet identified a single, methylation marker approach that would be predictive of type 2 diabetes. And certainly for mechanistic investigation, this would not be where we would want to stop our inquiry. So to get around this, some of the things that our group has proposed has been to set up studies that are a little bit more specific to cell types. One study has been a cell-free DNA methylation study as a biomarker for beta cell death in type 2 diabetes, and I'll explain that in the next slide. And another approach has been to look at single cells and their chromatin accessibility and transcription. So to look at epigenetics and downstream outcomes within single cells. So we are examining a new way to measure beta cell function or I should say beta cell health, maybe, by combining two biological concepts. One is that beta cells that die release their nucleic acids to circulation, and we call that cell-free DNA. And the second is that cell-free DNA from pancreatic beta cells can be identified by a unique DNA methylation pattern that is specific to endocrine cells. So here you can see that what we're trying to capture is cell-free nucleic acid that comes from the nucleus. And then here, I'm showing here, each of these are clones of, so like gene promoters, and these are different CPG sites. And if it's a dark circle, it's a methylated CPG site. If it's an open circle, it's unmethylated. So you can see that the insulin gene is primarily in the promoter is primarily methylated in muscle, liver, kidney, blood, all these other factors, and pancreas, because pancreas is mostly exocrine, not endocrine. But when you look at the islets, that these CPG sites are unmethylated. And this is there were animal studies and some human studies to build this as a potential point of further investigation. And so we jumped into this. We wanted to know if cell-free DNA methylation can be a biomarker of beta cell death. And so our goal was to recruit youth ages of 10 to 17 from Children's Healthcare of Atlanta to assess the discrimination of this biomarker, and also to examine how it may be associated with other clinical signs of diabetes risk or severity. And we included healthy controls, and we included high-risk youth who had obesity and they were at high risk and had family history of diabetes. Our DNA methylation methods were to take cell-free DNA samples and to have them be bisulfite modified and then amplified using PCR. And then the libraries were prepared using a special method by EpigenDX, which was a company that we collaborated with. And our key measure was percent methylation at single CPG sites. We actually had a very targeted list of which CPG sites we were going to look at. We examined 15 CPG sites from 3 genes that, after literature search, we found were uniquely unmethylated in islet cells, in the insulin gene, the glucokinase gene, and the amylin gene. And this is just a quick table for who we recruited. But you can see, as expected, the high risk and the type 2s were obese. The age was somewhere, around 14 years of age. And we had more African Americans in the high risk group and in the diabetes group. And so our results were that there were 2 CPG sites that their methylation levels significantly were associated with type 2 diabetes or severe prediabetes by linear regression. And when we did an area under the curve analysis for these 2 CPG sites, the AUC suggested that the percent methylation improved the discrimination of type 2 diabetes or severe prediabetes. We lumped together type 2 and IFG-IGT, combined IFG-IGT together to increase our sample size. That when you add the CPGs to the model, that it improved the AUC by nearly 10% beyond what age and BMI alone AUC was. So that was a really positive finding, but there was a very interesting observation as far as the directionality of methylation. There was more methylation found in those with type 2 diabetes, which is counter to what we were expecting, because we selected these with the idea that there would be less methylation for these CPG sites. And so, well, I shouldn't say luckily, we actually, instead of pulling my hair out, I was able to find two papers that have reported similar results. And it is possible that during type 2 onset that there may be other non-islet apoptosis occurring in larger concentration than the amount of islet cell apoptosis. So there's a really tiny amount of DNA that's being, released from these dying beta cells, really, really, really small amount. And that signal could be overpowered by something else. And what we're wondering is if there's active apoptosis occurring in type 2 diabetes that would be related to inflammatory processes or inflammation. So this is going to be explored a little bit further in our next study, but very interesting results that I think could potentially still be useful from a biomarker standpoint. So as far as now shifting into kind of another study I'm going to share with you, and this is one of our larger studies now that we're implementing, where we're looking at type 2 diabetes pathogenesis and thinking about inflammation, which has impacts on the islets, but also systemically has a lot of implications. And so obesity and systemic inflammation are intimately related to disease development through immune cell abundance and function. Oxidative stress resulting from inflammation can contribute to inducing modifications in proteins, impacting protein activity, stability, subcellular distribution. And the result of this could be a loss of transcription factor activity. And so we are looking at contributions of blood-based cell types. So PBMCs in diabetes are commonly described within the context of inflammation and disease pathogenesis. And there are a whole bunch of different cell types that we are looking at individually, but T helper cell types specifically coordinate the pro and anti-inflammatory action in type 2 diabetes. And so this is what is building into our active project that is supported by NIDDK and Emory, the Genie Center, and CHOA. And this is to interrogate the role of epigenetic control of these cell types specifically and their gene expression. The Th17 cells and the Treg cells, these are all T cells, they actually have this axis of balance. And that's what we want to explore. The Th17 cells contribute to inflammation by producing several pro-inflammatory cytokines that initiate and propel processes of inflammation, including IL-17, IL-22. I'm sure you've heard of these. Tregs maintain resolution of the inflammation by producing and secreting anti-inflammatory immunosuppressive cytokines like IL-10. And so these cell types are balanced in the healthy state, and it has been shown in certain conditions that that their balance is dysregulated. Also, what we do know already is that the differentiation into specific T helper cell types is regulated by epigenetic control. So we do know that there's this that the transcription of the RORC gene, that gene is a master programmer in supporting differentiation and activity, and that for FOXP3, that gene also controls differentiation activity to Treg cells. So knowing all these little bits, we are proposing this study, which is to map integrated single cell chromatin accessibility with single cell transcription in pediatric obesity and type 2 diabetes. And our objective is to identify the regulation of these inflammatory pathways and to use innovative methods in cell biology, specifically integrated single cell gene epigenetics and transcriptomics, to identify cell-specific gene networks. So our epigenetic technique is not DNA methylation, as I mentioned. It's actually this attack-seek. which is a method that identifies open chromatin regions where regulatory proteins can bind and provide information about how DNA is packaged within the nucleus. And what when there is more opening, when there's more accessibility, then those segments of the DNA are more likely to have transcription occur. So our aims are to first to compare. I guess I should tell you who we're recruiting. We're looking at type 2 cases, obese controls, and lean healthy controls. And these are all youth who are between the ages of 10 and 20. And what we are doing from their blood samples is we are looking at cell type abundance, comparing cases and controls. We are looking at the epigenetic regulation of T cell differentiation. So looking specifically at the chromatin accessibility around the RORC gene and the FOXP3 gene. And then we are going to integrate at the single cell level chromatin accessibility and RNA expression. I'm just boggled by this because I haven't done AIM2 yet, but I'm very excited to get cracking. Okay, so what we're doing is doing these 2-hour OGTs, getting blood samples, and then here's just a little snippet of how this works. But essentially, there's this transposition of, ATAC-seq relies on the transposed nuclei. There's a TN5 transposase enzyme, which it inserts sequencing adapters into open regions of chromatin. And the adapters allow for primer binding and PCR amplification to eventually then sequence. So individual nuclei are captured in these gems. That gems are gel bead in emulsion. And the DNA fragments and the 3' ends of the mRNA are barcoded. And so the barcode is pretty essential because that is what links to the individual cell of a person. And then ultimately what you end up with is 2 libraries from a single cell. So you get a library for the attack library, and you get the gene expression library. And this allows us to interpret epigenetic profiles with key expression markers. So we're producing ATAC-seq data on 20 cell types, over 20 cell types, RNA gene expression data in the same individual cells, integration of gene regulatory networks, key nodes associated with disease versus healthy state. And then we hope that this will allow us to identify new targets for mechanistic studies and new applications for diabetes prediction and risk stratification. There has been a bit of preliminary work. We are identifying the cell types, and we just finished doing the multiome assays for 16 individuals, and we're about to send out for those samples for sequencing. So coming back then to this, I would be remiss not to mention some of our other work that's related to identifying signals of beta cell decline, even though the focus so far has really been on kind of blood-based markers. But I do want to mention that we are actively developing studies in the actual pancreas for assessment. One of our studies is an islet function imaging study, which is very exciting. It's based on exploring quantitative magnetic resonance imaging, or QMRI, for spatially resolved in vivo measurements over the entire pancreas. And in our project, we're testing and benchmarking anatomic, compositional, and functional properties. I do want to emphasize the functional properties. This is functional imaging, like what's done functional imaging of the brain, but for the pancreas. That's what we're trying to develop. And to make this a tool that's sensitive to, it works as a tool because it's sensitive to the changes in oxygenation of endogenous hemoglobin. That's kind of the premise behind how this works. So there's no exogenous contrast that's needed for whoever you're scanning. And so, our preliminary results, we've collected imaging in about 10 people so far. And this is not from 10 people, but this is from our first person that we've been analyzing. Well, you can see T2 star, which is a measure of the MRI, that you can see that during a intravenous glucose tolerance test, which is what we're doing during the MRI, we're giving them an IVGTT, that we get a response in the bold signal, in the MRI signal. And so this is, so far, the results are suggesting that they are, operating in line with what we expect that we will see. We should hopefully have more results to share with you at the next time I talk with all you guys. And then lastly, I did want to share a project that we are doing with our partners in Hyderabad and AIG, Asian, oh gosh, I forgot what the I is, but it's our hospital for gastroenterology with our esteemed colleagues in Hyderabad who have really been launching this India Alliance project to develop and create the Center for Translational Research and Training on Endocrine Pancreas and Type 2 Diabetes in India. Very exciting project because the objectives here are not only to create a pancreas biorepository, but also to study the endocrine pancreas from a functional, cellular, and molecular standpoint as related to type 2 diabetes, and then also to allow for interdisciplinary training that will inform, hopefully the next generation of professionals in this area. And so that leads me to our conclusions here for the whole talk. I appreciate your attention. To find better targets for clinical intervention, there is a need to identify mechanisms driving disease onset and prevention, and prevent the wasting. We're just wasting, in the words of one of my mentors, Dr. Larry Phillips, wasting the phase where we could really make the most impact on the disease course. And population studies have been essential to expose the opportunities. And so it would be very important, while we are continuing to fund these mechanistic areas, to understand cellular pathways of type 2 diabetes, that the population studies need to continue to provide that information that informs mechanistic studies. The development of new screening interventions and new targets for mechanistic studies will yield new opportunities to address rapid decline of pancreatic islet function in youth, not to mention many other populations that have a similar fast rate of decline. And new markers that are more sensitive and specific will be important for us to develop. And single cell multiomic investigations, we hope will offer some promise as well. And improved indirect and direct assessment of pancreatic beta cell function at early stages of decline will change the prognosis of youth with type 2 diabetes. That I feel very strongly about based on the data that I've shown you. I would like to thank all of our all of our sponsors and supporters of this work. And I have to do a plug for our studies. So if you're interested, if you're a youth or a youth that would be interested in mapping that, doing the single cell study that I just shared with you, here is the contact information for the study coordinator. And if you're interested in supporting or being a part of the functional MRI study that I showed you, where we're scanning individuals with their IVGTTs, here's a QR code that you can use to sign up and fill out a little form to assess your eligibility. And I think that's it. Thank you so much.
Thank you, Lisa. We've got a couple of questions in the chat, and I'll read them quickly. The first one is from David Ryder. To what extent do we know if the emergence of microvascular disease could be a driver of diabetes progression in youth, in contrast to a complication that is caused by diabetes?
Sorry, can you say that again?
To what extent do we know if the emergence of microvascular disease could be a driver of diabetes progression in youth, in contrast to a complication that is caused by diabetes?
Oh, right. Yeah, it does go hand in hand. And I think if you talk to experts in other fields, nephrologists, hepatologists, etc., the perspective will be different. So there is certainly microvascular disease that is contributing to the onset of type 2 diabetes. And for some populations, that would be very important. Just thinking about liver disease specifically, and where it's in such high prevalence in Hispanic populations of youth, and where there may be more of a primary disease occurring from non-pancreatic driving that progression. So that is an important perspective. But nonetheless, once you get diabetes, you're prone to getting other complications, whether you got it first or second.
Okay, the next question is from Herman Taylor. Did the studies that found different optimal cut points for the diagnosis of diabetes and pre-diabetes test sickle cell trait?
We did look for that. I would have to go back and check, but I think we did try to exclude for hemoglobinopathies.
Okay.
But I don't remember all the details of what's available in NHANES for that, because we've done analyses on this area as well at the VA. And I do know that at the VA, we were able to exclude individuals with hemoglobinopathies. I think we did something similar in NHANES.
I raised it simply because sickle trait is often such a hidden and not thought of issue. For years, we've been sort of pushing the notion that it's a non-disease, but of course, sickle trait can be associated with lower hemoglobin A1C for a given level of glucose in a given individual, and be, if relied upon, be a source of delay in diagnosis or missed opportunities for prevention of sequela down the line.
Yeah, that's a very important point. And as we explore mechanisms, because now the next batch of studies, at least that we're starting to get involved with, is understanding a little bit more mechanistically what might be driving either high HGI or low HGI. Certainly, that would be an important reality to consider.
Okay, and the last question is from David Ryder. Is it possible to perform respirometry on the T cells you're collecting?
That is a good question. I don't have the answer to that, but it would be worth identifying an answer. That's a great question, David. Thank you for that.
Yeah, just a quick comment because I know we're out of time, but fantastic presentation, by the way. I really enjoyed that. Yeah, just the thought is that, if T cells are being affected by hyperglycemia, then it could have an impact on their ability to sort of turn into the H17 or the Treg cells, right? I think they differentiate from a progenitor cell. Is that the way that works?
Yeah,
So-
they do. Right. They would have
And
different
so
pathways.
if their mitochondria are damaged by excessive hyperglycemia, then it seems like they might preferentially exist in a certain state. So that could be an interesting thing to understand. I guess I also wonder too if you would be able to look for epigenetic markers of microvascular complications in this study as well, because it seems like that would be a real good opportunity given the prevalence of microvascular disease.
We would be able to look at that from the standpoint of if there are genes that have been identified that have been associated with microvascular complications, then we would be able to analyze it in that way by focusing on genes of risk. As far as having outcomes on none of these kids have, to my knowledge, and we'll go back and double check, but I think based on how we're selecting these youth, they may or may not have complications. So we'll actually, some of them might, because we're not excluding by complication. So that might be something we could do. That is something we might be able to do, actually. Because we have full sequencing on them. So really any kind of discovery project is possible as long as we've collected some of the appropriate clinical outcomes in those youth.
All right. Thank you so much, Lisa. And everybody have a great rest of your week.